Okay, so good morning, everyone. Before we introduce our keynote speaker, we would like to give a very special welcome to Mrs. Shirley Lakey, who is joining us today on behalf of the William H. Lakey Transplantation Lectureship Fund. It is impossible to talk about the history of transplantation in Alberta and even in Canada without acknowledging the groundbreaking contributions of Dr. Bill Lakey. This lectureship series has allowed us to host a number of fantastic, renowned keynote speakers over the past few years from both within and outside of Canada. Dr. Lakey performed the very first kidney transplant for Alberta in 1967 and would go on to perform nearly 800 more transplants in his career. Dr. Lakey was truly a transplantation pioneer who bettered the life of countless Canadians through his passion and dedication. There are patients here today in our in-person and virtual audiences who both had Dr. Lakey as their own surgeon or who would have benefited through his mentorship of the next generation of clinicians who took up the torch and continue his work. His family, including his wife Shirley and his son Jonathan, established the William H. Lakey Transplantation Lectureship Fund as a legacy of their husband and father's illustrious career and life's work. We are honored to have this type of support and to see Dr. Lakey's memory and legacy live on all of the researchers represented here today, who stand on the shoulders of giants that paved the early roads to transplantation that we build upon today. So thank you, Mrs. Lakey. To start our research day, I would like to introduce our keynote speaker, Dr. Alexandre Loupy. Dr. Loupy has made the lengthy journey from France to be with us in person today, so we hope you're able to meet and talk with Dr. Loupy after the session and throughout the day. Dr. Loupy is an active adult nephrologist and professor of nephrology at the Necker Hospital of the Assistant Publique Hôpitaux de Paris in the kidney transplant department. Dr. Loupy's research focuses on artificial intelligence and multi-organ transplantation analytics. It covers allograft transplantation, rejection, antibodies, and population sciences. He has 2 PhDs, one in cell biology and the other in biostatistics. He has authored more than 324 publications, holds 5 patents and software protections, and has given access of 32 invited international conferences talks since 2006. Many here today may have heard him speak recently at the Canadian Society of Transplantation annual scientific meeting in Banff this past September. We would like to give a warm welcome to Dr. Alexandre Loupy, who is bringing us our keynote address today entitled Multidimensional Approaches in Solid Organ Transplantation, Insight for Diagnostics and Prognostics. Welcome, Dr. Loupy.
Okay, you sure? Yeah. Yeah, so really happy to be here. That was kind of a journey to reach out to Edmonton, but went really smooth. Thank you a lot for the support, the organization, for the invitation. As you know, that the first time I came to Edmonton was back in 2011 when I was doing a short stay in the ATI AGC lab with Phil Halran, and I met Michael and you, and so it started more than 10 years ago. Really happy as usual to be here. I would like to thank Laurie also for the very kind invitation and a very productive day we had yesterday and a lot of exchanges, nice dinner, amazing. So my talk will be really to start to think about how, you know, we can use tools for clinical decision-making process. And I don't really like the terminology of AI, but how algorithm actually can help into the way we manage patients and how we can see the implementation of those tools in the present and the future. So that's not going to be about science fiction. And I wanted to start with a slide, and you might recognize here. Wait, okay, I'm good. Yeah, you might recognize someone in the audience here. And that's a quote from Michael. And, you know, so Michael and I, we are good friends and colleagues. And, you know, I like when Michael says that, oh, Alex, I mean, your team, you're doing so well, amazing research. So you're so happy. And they say, but then you're waiting for the next part of the sentence. But do you have any plan to make it useful at some point? And so, yeah, you're not that happy when you hear that, but at least it's very interesting because first of all, that's a really valid point. And point number two, it pushes us towards thinking about, okay, it's good to do science, it's good to publish paper, but we also have to think whether those paper and that research can ultimately translate it into patient care. And that's a gigantic step to achieve. And we are maybe not there yet, but I think we are making progress, because really the unmet need is how we can better predict and tailor the risk of rejection, graft failure, but also the risk of cardiovascular complication, infections, cancer, and the risk of death in kidney transplant patients. But that's true in all medical fields. And also talking about allocation, what will be a utility of utility index when you have a patient and an organ offer, and how you can use simulation algorithm to actually build some kind of digital twin and project that offer into the likelihood that the patient will benefit from a transplantation. So that's really important. And I'm not going to make a big deal about AI and the promise. I mean, you've heard like thousands of YouTube talks and everything, even on Netflix. And the recent ChatGPT stuff, which is amazing for text mining. So there is a lot of promise, but we are talking about healthcare. And at the moment, we also have to think about the danger zones of AI. And you're aware of this huge initiative by IBM Watson in the past year and the healthcare. Basically, what they did is that they were using electronic medical records in oncology, and they built some prediction algorithm for response to therapy. They were using an army of people, biostatisticians. They were doing sophisticated computational, methodology, and at least very politely, some people made some, let's say, comments about the usefulness of the tools that IBM Watson was providing, because that was really the point where they had such a mass volume of data, but they did not have the quality of the data. They did not have the velocity of the data. They did not really qualify their data. So they really relied on unstructured and low-level phenotype data set, and which were not designed to address the question. So they had a volume of data, but they did not have actually the appropriate data set to answer that specific question. So it appears that their system was not generalizable or transportable to different clinical settings, different cohort, and different clinical scenario. And the algorithm was making wrong predictions, which ultimately could harm patients. So we have to be very careful about that. And more recently, you've seen this maybe epic AI-based sepsis prediction model published on like a huge amount of patients, not validated, sophisticated methodology, and a crappy output, actually, because this JAMA Internal Medicine paper showed actually that the clinical use of that algorithm was actually provoking more harm to patients than benefit. So it was absolutely not externally validated, and the external validation process was not done, and that was not good. So I think we have to be cautious about those examples, but you have multiple examples around AI. So let's also not be too depressed, but this is like a figure that I did recently to illustrate, how many models and prognostic models you have. And it's not only in transplantation, it's in overall medical fields. And the Leaky methodological for implementation in clinical care, because the models are not fit for purpose, they are developed on wrong or biased population, they are expensive, or they have non-available predictors. It's time intensive to use the models, and the outcome are measured unreliably. It's hard to pronounce. So it's also the point where registries are used to make a lot of models and predictions. So I think registries are useful, but they are not designed to address prognostication in a way that all prognostication models built from registries in transplantation, like what KDPI, KDRI, but other prognostic system, they are not performing really well. Point number one, they are not calibrating well. Point number two, and they are not really able to show transportability and stability. in different contexts and scenarios. Most of those models are not properly validated. You can call external validation, but when you make a deep dive on the methodological appraisal of validation, you see that it's a kind of low-hanging fruit of validation or external validation, because those external validation cohorts are not really external validation cohorts, and the models are not appropriately validated. So it means that the models cannot be implemented because they do not show impact on decision-making or patient outcomes. There is no dedicated software developed. You know, a Shiny app is good, but from a Shiny app to a compliant software that got the CE mark, which is a clinical decision support system, there is a giant, you know, step. And also you have to require for adherence to regulations, which is also an important. So it appears that most of those models are not adopted because the prediction appear to be not useful, prediction are not trusted, and the model are not transparent enough, and the tools are not really available for clinical practice, and there is no really good study supporting the full proof of clinical utility. So yes, we have to see maybe the glass half full rather than half empty, but there is a long process and a long roadmap towards implementation of you know, models in clinical practice. So let's not be depressed. There are ways maybe to advance the state of the art, and maybe, you know, transplantation is seen as a tiny specialty. You know, and I've been struggling, and Laurie as well, I mean, all of us, when we apply for big grants, you're always competing with neuroscience, cancer, cardiovascular diseases, and they say, okay, what you do is great, but I mean, oncology has so much impact. And I disagree with that. You know, it's just, I think our specialty has a lot of potential to have very impactful and proof of concept discoveries that can be applicable and transportable to all different specialties. So we should be proud of the transplant field, because it's an unprecedented opportunity to have very well phenotyped cohort when you can build database system, prospective and selected cohorts. That's what we try to achieve in Paris for the past 10 years. We have invested a lot of human resources and money for data quality, and to have, you know, an integrative research strategy where every single patient that enters the network is the subject from the day of transplant until the day the patient, you know, loses graft or dies, to every single medical information, every single biopsy, every single treatment, every single modification of treatment, every complication. Everything is recorded in a structured database system, which has been designed to collect this information prospectively. And we build pipelines between platforms. We homogenize to have the most possible standardization. And we are also working with an army of research assistants who on a daily basis make those data, you know, accurate and qualified. So that this is part what we have developed in the Paris group for the past 10 years. And to end up with you know, those algorithms that we are now trying to implement in clinical practice. But I would say that the fun part is to do the models, and the really complicated part is to build those data sets. It's very, I mean, it's a huge amount of work, and I'm really grateful to all the people involved in this Paris Transplant Group database system, which is still operating thanks to academic grants. So this integrative research strategy allowed us to have some different milestones. And this is just an example, or just to illustrate the systemic nature of the approach, because not only we're able to publish in specialty journal, but in more general audience journal. And we like that because it's to us the proof of the systemic nature of the approach, and the fact that the results might be interesting for a wider community than only the transplant field. So we have made progress, I think, on risk stratification, allocation, empowering clinical trials, moving into precision diagnostics, better address disease heterogeneity, connect, especially in the field of antibody-mediated rejection, the antibody properties with different phenotypes, different biology, different injuries, and different responses to treatment. In other words, moving into a more personalized medicine. So I will give you now 3 concrete examples on how we build systems that we are trying now to use for clinical trials or clinical practice. And I will be starting with prognostication, algorithm-based decision support for predicting long-term allograft outcome and patient outcome for clinical trials and patient care. And don't expect that to appear like in 6 months of research and you have a database and you do models. And when you talk about predicting long-term allograft failure, this is not easy. That's not trivial. And most of the literature actually try to always to connect biomarkers or stuff with long-term allograft outcome, but none of the studies were really designed to address that. So they suffer from some bias, which prevent from building really robust prognostication system. And I will be illustrating the iBox roadmap here. When we started to build the iBox initiative, wanted to build a very robust, transportable, and validated and qualified system that predicts long-term allograft outcome, and that could be used in many different clinical scenarios and cohorts in different countries. Started back in 2012, when we designed the iBox project based on those prospective cohorts. In 2013, we had the protocol finalization. 2014, we moved into the data curation plan, internal validation studies on biopsy, central reading, retesting for DSA in a centralized platform, longitudinal data acquisition. 2015-16, we started the statistical modeling. We generated the first scoring system in the development cohort. We started and engaged the discussion back in 2017 with the societies and agencies, including FDA and EMA. 2017-18, we spent a lot of time to gather a lot of external validation cohorts and RCTs. We did post-hoc analysis on those RCTs to see whether the iBox system was fitting well or performing well in those different RCTs. So we had data acquisition. We work with industry to have the access to all these data, and we did a huge data curation plan for those external validation cohorts. So cohorts enhancement brought us to 2020 to FDA letter of intent, and EMA further refinements on cohorts and studies. We published the seminal paper in BMJ, and we engaged discussion with ESOT and AST, started a partnership with TTC, the Transplant Therapeutic Consortium. After the paper was published in 2019, we went into the additional validation of the iBox system. We published the dynamic iBox in Lancet Digital Health, the first application in a clinical trial virtualization in BMJ, and Transform trial, and we had the EGFR trajectories published in Keynote International, many ongoing studies and papers that are the subject of ongoing submission. And in 2022, we were glad to obtain the qualification from EMA as an efficacy endpoint and surrogate endpoint for clinical trials. And the FDA is ongoing, but as you can guess, we have many difficulties and hurdles, because qualification is quite something for agency, even if the reasonably likely surrogate endpoint. So clinical use also was very important beyond the surrogate endpoint, and I'm going to be giving you some concrete example on how we are pushing iBox now in clinical practice and having a randomized control trial to evaluate its clinical benefit. And the recent addition in pediatrics and above automation that you've probably seen in published, just released in Nature Medicine. So, I mean, to make a long story short about iBox, we started with a very holistic approach in this database where we had all possible kidney transplant prediction or parameters which can be useful for predicting long-term allograft loss. We didn't want it to be fancy that nobody could use. And there is like no surprise. The parameters you see there are the parameters that actually matter and recapitulate the course of long-term allograft failure. You have functional parameters such as EGFR and proteinuria, scarring on the biopsy, presence, absence, level of anti-HLA DSA, GPDC, macrocirculation injury, INT lesion, CG, and time from transplant to evaluation. But the point is that that final model was possible because we were able to integrate the entire spectrum of potential parameters that can be encountered post-transplant, and the quantification of the respective amplitude and coefficients of those parameters independently showing independency, but also incremental value was important. And the features of iBox algorithm is generalizability, the specificity, because it has been developed for transplant scenario, the causality, because all the parameters you see there are parameters which actually really matter, directly related to the predictors of long-term allograft failure, accuracy, but also feasibility and transportability was really important. And, we were happy when we published the iBox paper, but this is where, my life became a nightmare because when you start engaging discussions with agencies, like they push you like really hard. And they were asking us like a lot of things, not only from the pure regulatory perspective, because we had to have all the agreement for, and we pushed all our data to FDA because it was the subject of data quality check and the models were challenged. But they were asking very fair questions, but every single component was taking a lot of time. So the wide validation of iBox is that, okay, they ask us, okay, we have to validate iBox in African American, non-African American, in patient under TIMO, in patient under belatacept or CA9 inhibitors, using EGFR and proteinuria instead of histology, using histology BAM scores or diagnosis instead of BAM scores in the first year, after the first year post-transplant, in living donors, in deceased donors, in patients with high degree of sensitization, non-sensitized patients, with different induction therapies and more than 38 scenarios were used to validate iBox. And we showed that actually the iBox was really stable among those scenarios, which is good news. The performance C statistics, but also calibration or Breyer index, which is a combination of both, was really stable. So we didn't have a system that is perfect. You're not going to see those like rock curves and the prediction which are perfect, but it's 0.81, which is really good. And also calibration is good and conserved. We were also challenged by the methodology. They say, oh, but iBox is just using a Cox model. We are in the 21st century where machine learning can outperform every single old models and old methodology. So we had actually to build an entire pipeline where we had to redesign and adapt the machine learning tools to time to events. And we were able to show actually that all the like fancy machine learning in that scenario for long-term allograft prediction were actually not adding anything. You're basically overfitting your model when you use those AI, or let's say machine learning tools, but the calibration is not as good. So the Breyer index, which is a combination of both, is lower. So machine learning in this setting doesn't add anything except the fact that you look fancier when you submit a paper. It's just completely useless in that scenario to predict long-term allograft outcome. We published that recently in Keynote International. Also, they say, oh, what about children? Please validate the iBox in children. So 3 additional years and efforts, and a very nice collaboration we have between 7 US centers, 2 French pediatric centers, more than 700 patients, kidney transplant recipients. It was really hard to get. That was a lot of effort, and you see all those sites participating to the pediatric iBox. We are not even expecting the iBox to perform well in a pediatric population because, IFTA, I mean, you're not going to give a marginal kidney to a child, right? But obviously, and that paper is currently in revision in HAT, we ended up with the same similar performances in the pediatric population, which is a good news, and that shows actually the kind of universality of the prediction system. That's the iBox validation in pediatrics. You can see the C-stat with the bootstrap confidence interval of the C-stat and the calibration, which is again really good. So I mean, the application of this algorithm becomes clear for clinical trials. Industry is not investing our field anymore, or very few, which is too bad. And I can understand because the quest for the hard endpoint takes 8-10 years just to be able to show the events. And when you use BPAR, we know BPAR is like really bad surrogate endpoint. So it comes to be used as an emerging surrogate endpoint. And I'm going to show you some results. So this is, for example, Rituxera, which is a trial, a randomized control trial that, and actually you can see the published results of this trial, very few patients. It's a study on antibody-mediated rejection. But so they randomized patient at time of AMR, they give treatment A versus treatment B. So it was the comparison of PP-IVIG versus PP-IVIG plus rituximab for the treatment of AMR. They did not show any difference, and they had to wait for 7 years to show those Kaplan-Meier curves. So I'm going to show you what you can get with iBox, but only 3 months after the randomization, after the treatment. This is the actual curve that you can see, which is the projected, survival by iBox. And as you can see here, do you see the arrow here? No? No. Okay, no. On the screen? No. Okay, so no problem. As you can see here, 3 months after randomization, you would have had the answer at 7 years. So this is a context of use of iBox in the randomized control trial. You know, you can, at a very early stage after randomization, use iBox. And because it has been validated in also many randomized control trial and different scenario, you can make the projection and anticipate that the regulators will make some conditional approval on that. We are not there yet, but we are taking, I think the medical field is taking that path forward. Let's look at, you know, patient with T-cell mediated rejection. So this is a study that we use in a large cohort of patients in our institution treated for T-cell mediated rejection with steroid pulse and tapering. And you can see these observed allograft survival up to 8 years after the treatment, which is the blue, you know, Kaplan-Meier curve. And the red dots is what you can get only 3 months after the treatment of TCMR in terms of prediction. So as you can see, you fit perfectly well with the true outcome that have been observed. So then you can see the potential of iBox in those scenarios for clinical trials. But you might say, okay, it's good in a clinical trial to think about graft loss. It's also important that, you know, you can also study patient mortality. And we are currently working on a mortality iBox, which took us an additional 5 years, because as you can guess, the determinants of graft failure are not the same determinants of patient death. You have to take into account a wide array of potential parameters that might explain patient death. So we are working really hard on that. And I hope I will be able to present you some data soon when the paper will be published. So we've seen for, you know, clinical trials, but what about patient monitoring? Because you can think about how the prediction made by iBox can help you in your decision-making process. So that's a more difficult part, I would say, because you first have to convince clinicians that an algorithm can outperform them in terms of risk predictions. That actually concern pushed us to design that study. From the Paris transplant group cohort, we randomly selected 400 patients and we built a dashboard for physicians. And in that dashboard, you have all the parameters that you will dream of to predict long-term outcomes. You have, you know, day zero parameters, donor recipient, past medical history, DSA, biopsy, function, proteinuria, everything. And we asked the physician at that time to make prediction up to 7 years, whether the graph would be failing or not. And we compare that with the computer system, which is iBox. That's a paper we published recently. So the data were kind of compelling. So we, as the physicians, and there were 9 of them at different stages of their career, they were not able to make good predictions. Their reproducibility was bad. They were not using the same parameters. So the C-statistic the discrimination was 0.65, which is not good. Calibration was really bad. And as you compared with the iBox system, of course, there is a huge difference. And a very interesting also information from that study, and we try not to promote that too much, is that the senior and the full professors were not doing any better than the assistant professors, which were doing better than the residents. Oh my God. And, you know, I'm a full professor, so I'm in that basket, you know, of not doing any better than the assistant professors. So, you know, if you make the hypothesis that when you have a patient in front of you, you are processing, you know, you're using your experience, you see, you know, the blood, you know, the past medical history. And if you think that risk stratification is important for decision-making process, for giving the right treatment, you might also think that that kind of tool can help in the decision-making process. That's not a proof, but that's a hypothesis we can formulate. And actually, to answer that question, we designed this randomized control trial because iBox is now embedded in software, which is CE mark. And we have this Predigraph software that clinicians can have. And patients can also benefit from that. They have interfaces, so they have an OCR, so they have the apps. So when they are out of the hospital and they they have an exam, they can screenshot, the screenshot suck into the into our database, align all the biology and calculate iBox and see whether the patient deviates from its trajectory or not. And then as a physician, you have an automatic alert or not if something is going on. And with that alert, you can decide whatever you want. You can discard the alert, you can say it's crap, you can say it's important, you can say, oh my God, I'm going to do a biopsy, or I'm going to do some cell-free DNA, or I'm going to test for DSA, whatever. So it's important because it's one of the first randomized control trial where you compare for decision-making process a physician enhanced by the machine to the physician using the standard of care parameters. We have recruited so far 500 patients in France and Europe, 13 centers. So UK, France, Spain in Barcelona, we have Switzerland, we have Israel, we have Germany as well. And the primary endpoint is how an iBox automatic alert will be handled by the physician and how a physician will, change the treatment or perform a biopsy. And if this enhances the yield that the biopsy shows a clinically actionable information. So those are the endpoints. We also have economical studies on that. And it's 18 months recruitment. The recruitment is over. So I guess that in less now, but 18 months, in 12 months, we're going to have the final results. We're going to see. Maybe the answer will be that it's absolutely useless. Maybe the answer is that it's useful. Maybe it's going to be in the middle. We don't know. But at least it's important. Just to let you know that it's not nothing, because this Predigraph software is reimbursed in France. We have 9,000 patients using it in different centers. So we are confident that we can, promote the use of that system. And that's the first part. You can think about a bigger dashboard with many different informations when you had biomarkers and cell-free, or, and all the typing and the stuff on ABO incompatible that we were talking yesterday. And according to the context of use, the algorithm will tell you informations. So what about talking about biomarkers? What about biomarkers? And we've been working quite a lot on the properties of antibodies to bind complement and how those antibodies can be, harmful for the graft. And there is the new kid on the block, the liquid biopsy donor-derived cell-free DNA, which came and was mainly studied in oncology. And we are working a lot, and we were able to gather big grants on biomarkers. But first of all, we wanted to have a deeper understanding of the transplant biomarker literature. So who knows who this guy is? Raise your hand. No? Jeff, No? You know who? Great. So Ioannidis works at Stanford, and he's like the really expert of meta science, the science that evaluates science. So he's a nice guy. A lot of people don't like him because he sometimes he says truths that we do not want to hear. But look, we have a good relationship. I was in Stanford in April, and we were exchanging about projects. So we are working together because I came to John like 3 years ago and say, biomarker is like prototype. In transplant is the prototype for specialty where you have a lot of biomarker study out. And you don't really know which are actually helpful and for what. So we say, okay, we're going to take every single kidney transplant biomarker study published in the past 10 years that associated the biomarker with outcome. And we're going to recruit a lot of methodologists with us, because assessing thousands of papers takes a lot of time when you have to make a deep dive into methodology, design, a number of patients, validation, adjustments, and everything. So we ended up with around 800 biomarker studies, and that's the study design here. This paper is currently submitted. First of all, we draw a landscape of the prognostic biomarkers in transplantation. That's the meta-epidemiological study. As you can see, you have blood-based, intragraft, urine-based, combination, other. Yeah, I'm not going to make a big deal about that, but that's important. And most of the biomarkers are actually blood-based biomarkers. No surprise, because everyone is trying to find how you can get rid of unnecessary biopsies, right? But you also have intragraft, biomarkers studies using, different technologies. Urine-based, of course, But that's the complete landscape of what we have in transplantation. So I think it's really interesting. But the most interesting part, and to make a long story short, because it took us like a lot of time, is that when you are looking into the take-home message, the median number of patients in those biomarker studies is 232. 77 were single center, 82% were retrospective. Only 4.7% of the studies performed true external validation, and only 3.1% of all the studies adjusted their biomarker on standard of care parameters. So one of the problems in the biomarker is that people have the tendency not to challenge their biomarker with the standard of care, and that makes your biomarker look good. If you take, biomarker X and say, oh, my biomarker predicts rejection and long-term outcome. Yes, but if you do not adjust from, for GFR, proteinuria, fibrosis, and a bunch of parameters, I mean, the likelihood that your biomarker actually makes it independently of that is unlikely. So the take-home message of that big study, and again, to make a long story short, is that most biomarker studies tend to be single-center, retrospective, with small number of patients and clinical events. Moreover, very few studies shared code and data, and hardly any were registered. So that's not really good news, but maybe we have to think about, so what will be the new biomarker studies and the way we have to design them. So this is what we tried to do. We applied for a big European grant a couple of years ago. And so my goal was not to make any assumption or any hope on the biomarkers. I just wanted to take all the biomarkers we know and which have been published in the transplant literature, and I wanted to test them in the real life setting of a multicentric prospective study, unselected cohort, and putting every single biomarker into the test of what we are using as physicians for our decision-making process. And as you can see here, we tested a lot of them, many platforms, and there is a lot of coordination and organization in this grant. You recognize a lot of them, urine CXCL9, 10, urinary mRNA, some blood mRNA, the endothelial targets, blood protein, blood and urine proteins, cellular-based functional assays. We also tested epitopes and all those stuff. And DSA was not like considered like as a potential biomarker that you would see, but DSA, of course, was tested. So study one, 950 prospective patients in Europe, France and Europe. None of the 60 non-invasive biomarkers show any independent and incremental value to detect rejection over standard of care. So that's the result. There is not a single of those biomarkers, and I'm not putting DSA in that basket, right? But none of those, biomarkers do not show anything incremental. So, the grant agencies was not that happy with that because they gave us 7 million euro just to show negative results. But I was like, no, come on. That's a really important result because it's not to blame those biomarkers. If you just make like univariate analysis, you're gonna see some differences, of course. It's not to say that the studies were wrong. It's just to say that the studies have a design where the context of use was not the clinical implementation of the biomarker, and that's the point. So I'm kind of proud of of that study, and we're gonna try to sell it to a high impact journal just to say, okay, look, it's very important because biomarker is not only in transplant, it's in our overall medical field. So the only one standing, guess what? DSA. Oh my God. In French, we say tout ça pour ça, all of these are just to say that DSA is important. But what about cell-free DNA? We wanted to have the same approach with cell-free DNA. And I'm going to tell you like the story about cell-free DNA because I think, and we talked about that, I think, with you yesterday. So the point with cell-free, where I approach CareDx and say, look, we have this big grant and this big cohort. And we have the potential to test whether cell-free makes it, and or not. So it will require two things. First of all, you're going to be processing thousands of samples for free for me. Point number two, we have to sign an agreement, because that's a research agreement, whether whatever the results will be, we will be publishing it. And point number three, you will have absolutely no access to any outcome. You're just going to run numbers, sera, plasma numbers. You're going to send it the numbers. My biostatistician will de-identify. And those like people doing mathematics and stat, guess what? They don't care if it has to be associated, not associated, up, down. They don't care. They just do, the math. So we designed a study, and we had this, of course, the prospective cohort, and thousands of patients. And we have also now US and European validation cohorts. But that's the same design. And, for the sake of time, I'm not going to go through all the details of that, but just to give you the take-home message that we found no surprise that, cell-free DNA was increasing with diagnosis, especially, active AMR, acute TCMR, mixed rejection, chronic active TCMR and AMR, no surprise. And also you could see the increment with injury for G, C4D deposition, G, and PDC and CG. You could also see that for ITTI band scores. It's brand new to show that, but that was kind of expecting. But that's the most important result. When we challenged cell-free DNA with the standard of care model that was used for the Utrain 60 biomarkers, you could see that actually the two standing biomarkers were cell-free DNA and DSA. And cell-free DNA, interestingly, provided an independent and incremental value over DSA, even when adjusted on function, stability, proteinuria, if you have dysfunction or not, you are stable or unstable, if you had previous episode of rejection or not. So I think it makes the point pretty clear that if you have a clinical decision model and the discrimination of a model with or without cell-free DNA, you don't have a perfect model. You don't have an AUC of 0.99. That's impossible. But at least you can see that the integrative IDD-cell-free DNA score provide an incremental discriminating capability, and the score also shows good, calibration in derivation. Of course, derivation, cohort calibration, it makes sense, but also in all the external French, European, and US validation cohorts. And that's the currently integrative donor cell-free DNA scoring system that we have built, where you enter the parameter, and that's going to project your patient into a likelihood that your biopsy will show clinically actionable informations. Interestingly, we think the same for molecular diagnostics, that we have to get rid of just one platform. We have to move into decentralized platform for cell-free DNA. And you know that CareDx has this technology, which is called AlloSeq, and you are using only 200 SNPs instead of 400. And you can, on site tomorrow, especially in the HLA lab, you can build your own cell-free DNA platform and test the cell-free DNA with their technology. Good news is that the price is like 600 euros and not 2,700. And we have built the platform, all SOPs, and we have everything in place. The platform is operating. We are processing samples from many centers because given the results we had, we started a randomized control trial to test whether if you are using cell-free DNA compared to the standard of care, will you be able to really reduce the number of biopsies or protocol biopsies and unnecessary biopsies, or can you trigger a biopsy that will show something? So this is part of the trial we are currently running. We have a randomized control trial. We have included 400 patients, arm 1 cell-free DNA managed with the score IDD cell-free DNA. The other is standard of care. But the answer we wanted to get is kind of European centered, because we are doing a lot of protocol biopsies for every single patient. 3 months, 1 year, we do protocol biopsies to everybody. So the study was designed to show that using that, if you could cancel those protocol biopsies, and we have an economical study, and we're going to get a reimbursement in France 2024 if the results of the randomized trial is positive. We will see. So just to illustrate in biomarkers the fact that actually DSA and cell-free DNA looks like to be good biomarkers for patient monitoring and ultimately improving transplant care. So I'm going to end up with that, and I'm not going to spend time here because I think you had many talks from Phil and Michael about precision diagnostics in biopsy and how you can enhance that. Of course, I think that the molecular dimension allowed, first of all, to better tackle some overlapping pathways between the manifestation of antibody-mediated rejection across transplant organs. You know that there is a consensus panel using FFPE-based technology, which is called the BHOT panel, which is the Banff consensus panel, and many centers are using it nowadays. The system, we think, has to be decentralized, because if you have a nano machine, and you can from all the studies we have generated scoring system, and we are building the ICDOT, which is the consortium in which you have access to classifier. It means that if you have a nanostring machine here and you have one, you can just run samples and you can log your RCC files, go in through a pipeline of normalization and step by step, and get molecular reports automatically by just connecting on the consortium. So it's like an open source and shared platform of algorithm where you can decentralize precision diagnostics. And what about automation of diagnosis? And I'm not talking here about but so the Banff rules became complicated with time, and you are lucky here to have outstanding pathologists, but that's not the case in the real world. It's really hard for pathologists handling kidney biopsy and heart and non-transplant and gynecology, urology, and cancer, just to integrate all the complexity of the Banff rules. So our hypothesis was that was leading to some errors. So we designed, and it took us 4 years, a companion tool where we encoded, decoded every single Banff rules, embedding that into an algorithm, and plugged that into a decision tree where we only build that system, but also validated. And it was interesting because we have so many ongoing clinical trials that we were able to show that there was like 40% of errors for the diagnosis of rejection in one way or another. So it was just released, so it's brand new. And again, the system is open access. You can just have access to the application and just use it. We think it's good for standardization, clinical trials, but also patient care. And it will help the Banff community to see what the strict application of rules will give you in term of diagnosis. So it's an easy to use system, again, open access, and it's really we are really proud that we were able to work with our collaborators and move that forward. So in conclusions, let's put that into the perspective of the cycle of A-hype roadmap. You have the technology trigger, peak of inflated expectations, throw of disillusionment, and plateau of productivity. So I guess we might start to be in the plateau of productivity after the disillusionment. And I'm talking about healthcare here. And I like that slide, but of course the doctors will not be replaced, but doctors who are using algorithm will likely replace those who don't. And I would like to thank you for your attention, and I'm taking also this opportunity to share with you the inauguration of the Paris Institute. So I saw the word institute here, so we are also scaling our team to an institute, which has just been granted. 4 organ specialties. We have 50 clinical departments and a staff of 80 people, 42 research team, 25 research units, and 4 universities. So I think I will have a more burden of admin work, but I think it seems very important because fostering research and collaboration is exactly what we want to do, and we'll be happy and open for collaboration with you also. Thank you for your attention.
Thank you, Dr. Loupy. We have time for 1 or 2 questions.
What a wonderful presentation. Thank you so much for going through all these things. Do you see the possibility of an iBox system for the other organs?
Yeah, very good questions. We are working on a heart iBox. So we are talking yesterday about what we call AI-CAV, the trajectory of chronic allograft vasculopathy that you can get. Actually, yeah, so we are working heart. Lung is complicated, but we are trying to work with Foch Hospital and liver to achieve that. Really, again, the quality of database. So to set up the heart transplant database, it's 10 people working full time. with the existing pipelines between HLA and platforms and everything. But now the database allows to start building a heart iBox. We started with the CAV, and I think Sinai is using in a clinical practice. It means that during the COVID crisis, they were able to use the algorithm, and in case the algorithm was predicting a likelihood to have a non-progressor in CAV, they just cancel the angiograms. Yeah, so they are using it in clinical practice. Yeah.
Thank you. We've got a question from our virtual audience. Simon, can he speak or? Okay, cool. Simon, where are you?
Can you hear me?
Yes.
Excellent. Thank you so much. That was a fantastic presentation and fantastic work, Alex. I'm still thinking a little bit about your model of the comparison between the physicians and the machine algorithm. And while it's pretty obvious that if you have a limited data set, that the machine will be better to analyze that and get details out of it, don't you think that in reality, kind of the repeated dealing with the same patient and thought giving over and over again may play into that as well? And in that context, I was wondering if you have any data on the time spent by the people and if that was lower in the more experienced people and maybe the background to why they didn't outperform the less experienced people.
Yeah, very good point. For the time spent, we have actually measured that because when the clinician was logging into the dashboard and we had the information about how much time he was spending. But I don't have the number on the top of my head, but you're right. When you ask a physician, and they did not do the 400 patients in a row, it took them months, so they were logging. You might expect that there might be a bias, which means that you do not have a patient. You're just kind of playing a game in a software just to make a guess. And maybe when you have a patient in front of you, you're not going to make the same predictions. I don't know. And when you have all that knowledge of the patients, so it's not a perfect study, but I think it's also illustrate that the predictions we are making are not that good. And I think that's the take-home message. We can improve that, I think, but the machine performs really good.
Okay, thank you. Please give a warm thank you to Dr. Loupy for this terrific and thought-provoking presentation.